students that may join later. Nowadays, okay. Can everybody hear me okay? So, welcome. This is a first of a kind fall 2018 Health Data Analytics Workshop. It's organized by the Michigan Nutrition and Obesity Research Center, specifically the Bioinformatics, the Integrated Bioinformatics Core, which SOCR, our lab, is the key component. I do want to acknowledge that we are funded by several other initiatives. Here is the MNORC initiative. We're funded by the National Institute for Nursing Research for the P20 grant. We're funded by the Parkinson's Disease Grant Center is acknowledged here, as well as the Alzheimer's Disease Center and several NSF grants. They're all co-sponsoring. Here is the website for the University of Michigan Alzheimer's Disease Center. Hiroko Dodge and myself direct the Data Management and Biostatistics Core. This is the MNORC website. You can find out more about it from the homepage, and then all the core services, including the biostatistics core, are listed here. And we have the Parkinson's Disease Center, which is a P50 grant from the National Institutes of Health. Is over here again. Part of the research that we're going to demonstrate was developed by all of these collaborations. So the website, how to find the website? If you just Google SOCR News, and we did ask everybody to bring their computers. You need that. It doesn't really matter which one you go to. Any one of these are all our websites. Go to one of these, find a date. And again, our motto for SOCR is it's online, therefore it exists. Everything that we've done as a team over the past 2 decades is, as you can see, fully available online in various formats. So for today, here is the link. I have it already open here. We just added the live stream, so and we're going to record it for subsequently if anybody wants to cross-check some of these things that we're going to be presenting. So the workshop is going to be is going to go through 3 different parts. The first one is the team is going to introduce the various SOCR capabilities. Then we're going to do some ad hoc biomed and health analytics challenges that you, as the attendees, want to drive. And there is a few that were already submitted. We're going to go over these, so that will be quick. And then we're going to break into small sessions, and I'll introduce the team in a second. We are going to kind of go around hubs. There is monitors, as you can see, around the room. We can share displays, whatever we need to do. This room is very high tech. We can do small sessions and talk and tackle specific problems that you may want to deal with. So the flyers, some of the logistics, we're not going to go. So today, myself, I'm Ivo Dinov. I direct the Statistics Online Computational Resource. I'll present the overall organization of SOCR and some of the analytics that we develop. Alexander Kalinin is a graduate student. He will present the Statistics Online Computational Resource Analytical Toolkit and some machine learning analytics. We have Simeone Marino, one of our faculty. He's going to present some of the analytics, including compressive big data analytics and the data sifter. Nina Zhu is a graduate student. Unfortunately, she has a conflict that she can't resolve, so she won't be here. But she works very closely with Simeone Marino on the data sifter, which is a very interesting tool you're going to see about obfuscating data that allows you to share it without compromising privacy or security of sensitive information. Saeed Hussein, sitting right up there, is another graduate student, an engineer, very bright. He'll talk about several projects, including data visualization, machine learning, and data dashboarding. And then Jerome Choi, right up here, He's a very bright stats graduate student. He will present one of the very specific MNORC case studies that we worked over the past 1 year, essentially interrogating fairly complex nutrition and obesity data set through some of the techniques that we've developed over the past couple of years. So there is a lot of links here, and they're listed so that you have them in a one place where you can kind of get to everything that's going to be shown today. You don't have to necessarily right now in this very moment track all these things, but I'll just point out some of these links. So the first one is obviously to the MNORC Center that I just showed you a second ago, and the specific bioinformatics core. Link obviously to the main SOCR website from where you can navigate to anything else. Now this is a link to the collaboratory site, which is also linked right up here. So we have a special folder that's specifically for this session. It's got a little README file. Have a look at this. Very, very simple. It kind of links back to the live stream, the G Drive, the website, you know. And then it has, so Zora, for example, is Zora here? Okay, Zora is over there. She's gonna be showing some of these driving, let's call them motivational challenges later. Jerome's project is listed here. If other people come up with stuff that you want us to discuss at part 2, remember there is 3 parts. During the second part, we wanna ask you to come to the podium and present certain challenges if you wish. You don't have to, but if you wish, so that we can kind of know where the audience stands in terms of the needs. Look, this is supposed to be, right, we disseminate and then you share the challenges, and then at the end we kind of tackle them together. That was the intent of this workshop. So that's the link right up here to the collaborative folder. And then there is a whole bunch of other things, right? SOCR navigators. We have so many resources. The most common complaint I get all the time is I can't find your resources. And obviously, I just showed you how to use Google search, for example, to find specific things like the SOCR news website. But we have some of these navigators that we have multiple different ways of navigating the SOCR resources. Okay, so that's not going to work because this little browser here does not have Java, but the other ones will work. Or Flash. Yeah, okay, so the browser doesn't have Flash, but these are JavaScript and they would certainly work. So for example, if you're looking for some of our ebooks, you know, you can kind of get right up here and double click in any one of these links. It's going to open up for you the corresponding resource. Okay, so you can kind of navigate. If people prefer hierarchical navigation, this is how you navigate some of our resources. There is a little bit more intuitive, right? So for instance, right up here under educational materials, you can go to, again, various books and double click in any one of these nodes, gets you directly to the corresponding resource. So there is hierarchical, there is structured, free, unstructured searches that you can do through Google, etc. So coming back up to here, there is a lot of data sets that in the absence of, if you don't have specific challenges that you have in mind, we have a very large collection of SOCR data sets. Look them up. Specifically, some of these I'll point out, the case studies, the biomedical case studies. So right up here, there is, many different things. World drinking water quality, divorce, quality of life, mental health, well-being and behavior. Here is Zora's data set. We just pushed it as a 28 case study, air quality. There's a lot of very interesting hospitalizations and a lot of interesting case studies that you can find here if you're looking for data. We have a number of electronic books, including, let's just see what do I get if I do a general Google search on probability ebook. So right up here, our ebook is whatever, number 5 on the list, just probability ebook. This has over 1 million users globally, as you can see. And this is just the table of contents. Now, each of these separate sections has its own users and et cetera. So we have some really powerful resources that we've developed. This, by the way, started over 11 years ago, but it is a very popular resource. We have scientific methods for health sciences, and obviously the predictive data science and predictive analytics. I'll show this in a second in a little bit more detail. We have a very interesting demonstration that I'm going to show you again in a minute about extremely high dimensional data and how you can deal with it. And let me just point out to you, some of our global users, you want to know, the other day I was presenting and one of my colleagues says, well, who are your users? And I'm scratching my head, trying to figure out how to answer that question, because who are your users is a very loaded question. So this is a Google map of the last 200 users in the past 3 or 4 hours of all the SOCR resources. They come from all over the globe. I don't know who these people are, but I can tell you precisely what they did, right? So this guy came from Sweden and looked at the F table. So we don't track. I know for a fact that in the US, about 6 or 7 years ago, I kind of delved, because you can go deeper here. You can actually find out what they did and okay. Oh, by the way, this is me right here showing you. See, I just toggled on here myself. So this is me. The last user is kind of me here going through the navigators in the Google map. At any rate, but about a few years ago, I did a little search, and I know that about 50% of our users are non-academicians. They're people in the industry, self-learners, informal learners, kind of the general community. But we have definitely over 15 million users over the past 15 years. So lots of users. Okay. Right, and we have some images and whatnot. So the specific services that we provide to collaborators, part of the MNORC, the UDAO Parkinson's disease, the Alzheimer's disease Center of Excellence, to some of these things are included over here. So we provide help with experimental design, modeling of preclinical and clinical translational research studies. We do a lot of randomization. Sometimes you want to do some kind of an experiment and you want to randomize your groups, your participants. We know how to do these things really well. We obviously know how to do power analytics and all kinds of other things, and we have apps for this developed. We do a lot of training of anything from undergrads all the way up to junior faculty. So we do a lot of these things. We help with HIPAA compliant data storage management, manipulations, and whatnot. We develop a lot of protocols for data analytics. And we, generally speaking, help with computation and statistical modeling, bioinformatics, anything that has to do with data-driven inference. So let me now, so that's the plan. We're going to go through these 3 parts. So I'm going to start first and kind of give you some of the things on the website. And Alex is in here now. So let's start off at the very beginning, socr.umich.edu or socr.ucla.edu. We have 2 hubs. One is at the University of California and one is here at Michigan. So just look over some of these things. We have a lot of apps now. They're not going to work on this. And by the way, this computer is just the worst computer to do one of these things on, because it's core image. And you know that none of you, I hope, has a core image machine that you work on, because these things are a lot. It's a can of worms. You can't do anything. But I wanted to. Okay, so she's got a core image. Okay, well, my sympathies. But I have my own computer that everything works on. But I want to demo it on a very light, privately controlled computer that has nothing to do. Now, some of my colleagues may actually plug in their own machines, because some of the high-end visualizations that we're going to be doing may require something a little bit more powerful. But anyway, so we have a lot of Java apps now. They're not going to work here, but if you have your Internet Explorer, for example, where you enabled, and if there is interest, I can show we have many, many hundreds of Java apps for simulation, distribution calculation, model fitting, charts and plots, literally many, many hundreds of these that we've developed over 15 years. The ebooks I already showed you, there is a link to all of these things. The navigators I showed you, the news, and now the web apps here I'm going to show in a second. So there is a lot, if you're interested in, and there is obviously this word, every one of these is a clickable link. If you want to know about publications, right up here are our publications. You can find out more details about some of our research, and these publications are very, very specific to the SOCR core mission. There is a lot of other ones, you know, including books and whatnot, many, many publications with proper links that you guys can navigate to find out what we do. So let me go ahead now and show, and here is again links to the data sets, some projects. If there are students that want to work with us, you can just do a Google search, SOCR projects, to find some of the projects that we have currently ongoing. Okay. Right up here, for instance. So you can look at various projects that we have ongoing. You can join any one of these projects, look at the descriptions and whatnot. So from the website now, I want to start by showing you some of the web apps that everybody should be able to run, no matter what machine you're on, directly in your browser. So, and we are, by the way, huge fans. The reason why we have so many users is that about a decade and a half, we made an executive decision in the SOCR lab to develop all the resources in such a way where we have client server. We are utilizing literally the power of distributed computing. We've done this 15 years ago. People started talking about cloud computing way later. So the SOCR essentially provides the instruction set to your local machine. Everything learns locally. We don't support services. You can't run on our servers. Well, you can, but for this you need actually special things that I'm not going to necessarily present. So, Alex is going to show, I'm not going to go into that. Alex is going to show Socrat in just 1 minute. Said is going to show the data dashboard. That's a really cute app here. Try to zoom in a little bit so you can see it more clearly. The PubMed Navigator. Let's look for Said, for example, Hussein. All right. So this does on the fly, and I'm not sure where Said is, but maybe we can. Is it SA Said? Oh, okay. Well, at any rate, so click in, and you can find out here. Clicking on anyway, you can actually traverse the publication record of people online. So this does dynamic on-the-fly query of PubMed. You know, this is incredible. You can take snapshots not only of you, your collaborators, their collaborators. You can kind of construct some of these networks that are useful sometimes to include. So this is dynamic and kind of list the publications. You can actually get to the publications depending upon, obviously, do you have. So right now, see, this is a Springer. Let's see if it records that I'm coming from the part of the University of Michigan. Some of these things, okay, so some of these things are not going to be accessible if you're not on the University of Michigan network. So I want to make this disclaimer, obviously. Should be obvious to everyone. Okay, so that's the PubMed Navigator. The motion charts is a really cool thing, and we have a publication on this. So this may be useful for many of you that are looking for analyzing data visually that's up to 7 dimensions, okay? So let me just show you in this specific case, and you can just look, I can do here Control-A, copy, and I can delete everything that's in here, okay? And I can just paste my own data. Just go ahead and literally you paste your own data. Once you paste your own data here, what you do is you go up here, bring the mouse over, and it allows you to map it. If you have time varying data, that's what you want to have under the key of year. It could be second, millisecond, you know, whatever. It doesn't matter what the measuring time point is. Then what should be plotted on the x-axis, y-axis? What should be the size of the blobs? What should be the color? How you want to categorize this thing? And once you have this thing, then you begin to you begin to interact with this thing. And see, in this specific case, we have defaulted to this data set because it's very interesting. For those of you, I mean, you guys are probably too young to remember here, but there was a 2007. I mean, 2007, it was scary. I'll tell you, lots of people lost jobs. People lost homes. It was crazy. I mean, in Michigan here, even California, the University of California, I think, had introduced twice in their 100-year span faculty salary reductions. And I was one of the lucky ones because we had grants, and they said, oh, okay, if you have grants, you don't have to cut your salary. Everybody else was cutting 8%. It was crazy, I'm telling you. During the housing bust, it was crazy. So you want to know, well, what's going on here? So in this specific case, we're looking at some economic. So housing price index is on the horizontal axis. And guess what? When the housing price index goes up and unemployment goes up, what is your guess for the next step? Unemployment going up and housing prices going up? That's a little combination. I can tell you what's going to happen right now. There will be a bust. It's very easy. But people didn't know that, right? So you can see some of these dynamics here and how you can explore. And obviously you can pause and you can exit. So just copy paste your data here. If it has something that you can track it over in terms of a motion, some kind of a longitudinal dimension, you select it, boom, you got your chart, and you can, again, the power here is not to generate a static image, because a static image means very little. But having it dynamically move across time gives you this perception of maybe effects that you need to model subsequently. So this is visual. You can model them subsequently using quantitative analytics that we're going to show you some examples of. At any rate, so that's a very, students just like you developed this. It was crazy. Nobody else, to this date, nobody else has a open source motion charts. We are the only resource that has that. Now, obviously, MindGap and various other things have the private versions of motion charts, but we have the only open source. Okay, so that's motion charts. Now, I should probably skip over this, but this is interesting for the if there are any people that are interested in non-parametric inference. How do you actually do randomized simulation-based inference on various things? So that's a very nice, again, we have a publication, students developed it. It's a great resource to do resampling and randomization-based inference. Violin charts, we will probably show some examples of this. We have some very unique tools that run, again, directly in the browser. If you're interested, so this is a three-dimensional representation of a two-dimensional Gaussian, and you want to know, well, what is this Gaussian? And now you can kind of start to introduce here. Let's say, suppose I want to elongate the Gaussian. I want to introduce some kind of a correlation. Suppose I want to have a correlation of 0.5, and maybe I want one of the axes to be slightly more spread out. And then suppose I'm interested in x, so it's between x, let's say between -infinity and 5 or something. And then if I start calculating, now look what's happening here. I can actually see the representation of what are some of these specific probabilities that are being computed here, right? So I can do on-the-fly representation both graphically and computationally of some of these things that I'm interested in computing, likelihoods of various things to be observed within certain ranges, again, here on the horizon. And now, does everybody see the elongated Gaussian here? Because I loaded it up with a little bit of XY correlation. And if I wanted to show it whole, right, it's gonna kind of look like this. So look from the top, right? So it used to be a perfect square. Now it's a little bit more elongated. Look at the changes of the axis. So we can do these things dynamically on the fly. Okay, so we have a number of very interesting probability calculators. These are, again, very unique. I believe we have the largest collection of, let's choose, I don't know, Cauchy distribution. So here is where we can kind of vary the parameters, and you can specify, well, which function am I computing, CDF or PDF, and then I can say, well, what's the critical value that corresponds to P to Q of 0.9, for instance. So I can do this on the fly. It says critical values 14.67. So if I know that I'm dealing with a Cauchy process and I want to find the top 10% of the observations, they're going to be exceeding 14.7 essentially. So we can do these calculations on the fly directly in your browser, nothing special, no licenses, no software to install. Okay. So Alex may mention something about the morphometry, the cell morphometry project here. We have a bunch of econometrics web apps. You can look at these if you're interested in economics things. Let me just briefly show you the brain viewer. This is something fairly unique, and Said is going to show maybe a little bit more on the brain viewer capabilities. But you basically can have any three-dimensional volumes of various formats. You can have shape information, and you can have other models, either atlases or tracks. And we have preloaded some of these, but if you drag and drop your data directly into that browser window, it's going to load them in the browser, just like I'm going to show you now, for example, for this example, right? So right up here is a interactive volumetric rendering of a real brain image with an atlas. And I can zoom in, I can navigate, I can scroll some of these planes any way I want, right? So you see how I can navigate that space. So here is my cerebellum in the back of the head, the vestibular ocular region of the brain. That's what keeps me standing up right now. And you can kind of navigate these things. If you're not interested really in cross sections or viewing the atlas, kind of the parcellation of the gray matter here, you can view this in fully volumetric rendering. You can change the opacity and pair right through the brain, and now you can, face recognition. Let's just see who is this Joe Schmo. I can see the ears. You can do a lot of things. You can make it kind of semi-transparent and go a little bit deeper, right? So you can see now the white matter tracks. Does everybody see the white matter tracks? The connectivity of the brain right up here, left hemisphere, right hemisphere. Right up here on the back is my cerebellum. So, and here is the corticospinal tract right up here. So you can do a lot of these things, including modeling. If you want to have tractography in the brain, or the volumes that you visualize don't have to be brains, but we've designed this, we've preloaded this with specifically with brains for reasons, because that's part of our secret sauce, effectively. That's jump too much. I'm trying to zoom out. See, when you display on this, okay, there we go. That's what I was looking for. So you see now I'm gonna triage. I'm gonna shrink some of the short connections in the brain so that I can see the corpus callosum that kind of connects your left and right hemisphere, right? And I can interrogate it and look at some of these. Red goes side to side, green goes up and down, inferior to superior. The predominantly blue colors go posterior in the back to anterior in the front of the brain. So I can actually examine some of these things interactively, and that's the brain viewer. Yeah.
What are the different colors?
The different colors in this specific case were showing the predominant direction of the tracts. People want to know, when you look at some of these tracts, the question is, are they, the corticospinal tract, for example, comes from your spine and goes through the limbic system, and then from there projections go onto the cortical surface, right? So the colors, the bottom to top, inferior to superior, are predominantly colored green. I know that these tracks kind of go up and down effectively. The colors that are predominantly red, they kind of go left to right hemisphere, and the tracts that are predominantly blue, they kind of go back to the front of the head. Now, this is you can color code them, for instance, by their length, by their how convoluted is the track versus how smooth it is. You can do various things, but you have some measurements on the tracts that you can, for example, the curvature of the tract. You can put some statistics, and then once you have the tracks for multiple subjects, you can do some analytics on some of these. Right? Okay. All right, so that's the brain viewer. Again, it's called the brain viewer, and you can paint. It allows you to really look. Sometimes when you have one of these solid objects, what you want to know is you want to parcellate it manually. In other words, you want to have, let's say, the hippocampus or other regions in the brain labeled so that you know you can compute their size. Well, the Brain Painter allows you to select a point in 3 dimensions and actually go ahead and have the computer kind of semi-automatically label it. The data sifter is something that Simone is going to show us in a second, a very powerful new technology that we've developed in the past year or so. The CBDA, again, Simone is going to show this. This Voronoi tessellation is a cute little app. Maybe if need be can show a little bit more. The autoencoder I'm going to show you in a second, or the autoencoder you guys can look at it. It's a very similar thing to what I'm going to show you now for this Tisney regularization, and then there's a whole bunch of other things. So before I switch gears and kind of show you the first example here, I want to ask if there are any questions or any comments regarding the overall structure and the type of things that we actually do in SOCR. So there is a lot of acknowledgments and all kinds of other things that you can see the team. Lots of students are involved. This is just right in here in the very same room a month ago. This was in April this year. That's the team about a year ago or so. Most of us are here. So, okay. So any questions or comments now before I start off with the first example, and then we're gonna go back to the regular agenda. Let me just see. So I need to close some of these because I opened too many. Okay, right up here. So Alex is gonna go after me, then Simeone, Said, and Jerome, and then we're gonna have a little break, and then we're gonna go into part 2, where we listen, you talk, and then finally in part 3, we're gonna kind of try to form smaller groups and see where we are. Questions or comments? Okay, so let me show you this. This was developed by another student, literally in the past 4 months or so. Now that's a very powerful tool that it would take a while to actually go through the. So I'm not going to go. So Vegi was the student right up here. I'm not going to go over the entire thing, but everybody should be able to do exactly what I'm showing you here. So this is intended as a learning module that I spent 2 hours in class. So we're not going to do that. But I just want to tell you that there is a nice motivation. There is an introduction. There is some details about the technique that's called T-distributed stochastic neighbor embeddings. There is a TensorFlow implementation, specific Python code for this. There is a video that you can watch just in case you're on a browser that is not powerful enough, the network is slow, whatever, you can watch the video. You can see the demo. We're going to do the demo now only so that I can give you a hands-on representation. And then I would encourage you all to try this with your own data. This is incredible. This is very powerful. And you can do exactly what I'm going to show you now with our canned data, which is certainly not a simple academic exercise. So this is the example that I want to show you now. So I'm going to zoom up. I'm going to zoom out so that I can kind of get the best of the real estate here. So let me tell you a little bit about this specific data set here. So this uses the UK Biobank data, and it does require a little bit of resources on your computer. And we have about 10,000 individuals. Every individual is a little glyph here, right? So for each of these individuals, we do have three-dimensional magnetic resonance imaging scans from which we've just extracted the middle sagittal plane to use as a glyph here. And I think I can, right. So here I can hide these things and kind of show you only the labels that are behind. So you see some people are labeled 1, some are labeled 2, some are labeled 3, right? So there's a lot of folk here. Okay? But what's more important is that in addition to the imaging data, we actually have 3,000 clinical assessment features. Now, what we did with these clinical assessment features, 3,000 dimensional data set, we augmented this with 5,000 imaging-derived metrics. For instance, the total surface area of the cortical surface, the volume of the hippocampus, the shape index of the cerebellum. So a lot of morphometry measures we've computed on these individuals, And now our original data, which you see has about 10,000 individuals, we went from 3,000 observed features to 7,500 features, from which we then did a dimensionality reduction to reduce these 7,000 features to 300. And now I'm going to act on these 300 features. The reason why we reduced this to the top 300 is that so that you can run this very complex data set that in practice requires many hours of high performance computing. You can run it in your browser right now. Okay, that's the only reason why we went down in scale here. And now here you see I can do PCA, directly principal component analysis, on the fly. It kind of shows me the three principal directions. As you can see, the axis, the red, green, and blue axes. And you see that there is predominantly one or two dimensions of variability here. But PCA is a linear operation. What I'm interested in doing is I'm interested in the very highly nonlinear approach of projecting this 7,500 dimensional data set into 3D using this new technique called TSNE, T-distributed stochastic neighbor embeddings. So this is now on the fly as I'm talking about detecting distances between the original data set in their native space, initializing the TSNE process, and now you're going to see the automatic, you're actually looking at the iteration of the optimization of a metric that quantifies that optimization process, something called Kullback-Leibler or divergence metric. The minimization of this metric you can see on the fly, and you can begin to see how certain, the topology of the cloud of data morphs as we go through the iterations, right? Look, and it's going to kind of reorganize itself, and at some point I may want to change some of these parameters, the rate at which the algorithm converges, is it supervised or unsupervised? I can also color code. If you have supervised, and in this case I have 3 labels, I can color code them so that I can see, do the red brains predominantly group in one region versus the blue, go blue, and the yellow brains, kind of go in a different region. So you can see these dynamics, and of course you can zoom out, and it's all very, very interactive. And at some point, you may wanna and you can sphericize the data. Simply means that try to project it on a sphere in a way, and see if that helps you a little bit with the interpretation, especially in 3D. So that's a very powerful experiment. If you would like to run this with your own data, you're going to be clicking on this link that's right at the bottom. Okay. And perhaps I can. Okay. So I need to be careful here because again, this machine is a flaky old dog with fleas. I don't want to push it too much, especially while it's computing here. Okay. So this is almost done. All right. And now we can see the evolution of this network. And at some point, you're going to stop this, and you can actually inspect various things, search for specific cases, for the cases 1 or 2 or 3, and you see how some individuals. And now my browser is definitely affected, as you can see. I can navigate because right now this is doing GPU computing 110% probably. I mean, I'm not exactly sure what the usage here is, but well, not okay. So this is only using 18%. So it runs on a single core in this specific case. And now, okay. So let me just pause it here for a second. And now look at this. I mean, look, the labels, you guys. These labels that we have are these. You can think of them as being clinical assessments. 3 different populations. The patients are red. The controls are blue, and the yellow are people kind of in between. And you can see a little bit of clustering, right? So the yellows tend to be down here. The red ones tend to be over here, not exclusively, but predominantly over here, right? And I can actually dive right through that cloud if I wanted to, right? And kind of see that it almost looks like a cloud that I'm kind of navigating through dynamically here. And then, so this kind of tells me right away that my data, my extremely high dimensional data, has some structure that can be exploited by subsequent quantitative analytics, right? So let me pause this now for a second, and I will click over here. And now, just to point out that you can load your own data, just save it as tab separated values. This is the tab separating the values. Does everybody see? The tab separating my values. So you can look at, this is the country data. You can just, let me just, what do? I open it in a new browser tab. Oh, this downloads it. And once I have it downloaded, let me open it with, oh, you know, I may have to, okay, so here's a trick that you sometimes have to do. So does everybody see the tabs that I have here? See, if I try to highlight, it goes all the way, right? It's not a bunch of spaces, it's a tab, right? So here is the data, okay, that I plugged in. And here is the corresponding labels. If you're working on a supervised problem where you have clinically or otherwise derived labels that you're trying to predict, you're gonna load in both of these. The second one is optional if you have some phenotypes, but this is mandatory, the first one. And once you have this, so in this specific example, I just have 100 countries, okay? And this is a PCA analysis of these 100 countries. And they are ranked based upon various criteria that I just showed you. You know, religiosity, socioeconomic, you know, mean income, the GDP per capita, you know, stuff like this is what's used to rank the countries. And if you sphericize them now, and you see I don't have now any idea about what's what here, but I can, you see, the zeros are, the ones are the countries that are ranked typically in the top 30 out of the 100, and the zeros are the other ones. And now I can actually start my t-SNE and begin to look at whether or not my red dots are going to be, let me color code it by the indicator. I want to see if my red clusters are separated from the purple ones, right? And at some point you're going to stop and you're going to say, look, you know, these guys, I'm sorry, the red ones are actually the 70 countries and the purple ones are the top 30. So you see, there is a little bit of separation here, and you can, if you want to know which one is which, you can actually go ahead and okay, so search, you can search for a country by name here. Okay, so Belgium and Belarus are right up here. You see, listed. So you can navigate. You can do your analytics at least visually in 2D or 3D if you prefer. It's up to you. Now that's a two-dimensional plot. You don't navigate anymore, and there is a bunch of parameters that you can vary. And once you do that, you know, various other things are going to show up. And because this is a highly nonlinear problem, occasionally you may get something that doesn't really make a whole lot of sense. Right? Okay. All right. So that's one thing that I wanted to point out here. So you can plug in your own data in this and see how it reacts. So that's probably a good place to stop for me and let my colleagues take it from here. Again, we are going to start with Alex, then go to Simeone, Said, and then Jerome, and then that's going to complete part 1. And then you guys need to begin to think about some of these case studies or use case studies, whatever you want to call them. Here was a genetic template that I wanted you kind of to look at. Here is Zora's, both the folder and the zip file and the HTML that we've done for her. You may want to look at this. You can drag and drop, just make a new directory here. And if you make a new directory, you can drag your data directly into our G drive partition that we have for this project. And then that's what we're going to do in part 2 and part 3. All right, so Alex.
Hi everyone, my name is Alex and I'm a 5th year now PhD student. I'm actually defending a couple of weeks at bioinformatics, so I'm working a lot with large biological data, such as cell imaging. I'm going to talk a little bit about the project, but also I'm leading a project called SOCR. It's SOCR Analytical Toolbox, which is focusing on interactive in-browser data analysis and visualization. And so we're going to start with SOCR first, and then I'm going to mention some of the other projects later. So we have a team of students who are developing this tool. It's all developed in-house, and there are a number of graduate and also a large number of undergraduate students who are contributing to the development of this tool. And the original motivation was that SOCR itself originally provided a lot of different Java tools, charts and modeler and analysis and experiments, and then many of them were very unique and interesting, and people were using a bunch of them. But then the problem with them that they were first not on a single platform, so each tool was separate from every other tool, and second, they were all implemented with the Java Web Technology, which is now by default is disabled to many browsers because of the security concerns, because every modern tool now is implemented in JavaScript. We are moving from Java slowly and migrating all those tools into JavaScript using SOCR. I'm not going to talk too much about the technical details behind that. We have, as Ivo mentioned before, we have a number of publications, and then we have a publication on SOCR design. So if any of you are interested in terms of large-scale web application development for data analysis and visualization, you can go check out our paper. Okay, loading now. We have a detailed description of all the technical underpinnings, what the technology inside, what kind of frameworks we use. There is a little bit of illustration how things work under the hood, so from when you load the data and what happens afterwards and what you can do with that and how you can implement a new additional plugin for SOCR or new additional module. So if you're interested in the technical details, please check out the paper. It's also on the website. I'm not going to go deep into those details. Instead, I'm just going to play around with the tool and see what we can do with this. So this is what it looks like, the welcome page. Basically, it implements sort of this simple, high-level interactive data analysis workflow. So you want to first, to have your data, you want to input it in some way. Then this main focus of this tool is tabular data, structured data that has rows and columns. However, we are planning on supporting other formats. But then, as you know probably better than me, that data in real life is never clean, is never well structured. So sometimes you need to what's called wrangle it. You need to fill out missing values or drop some columns or like swap values or split strings that you have to extract some meaningful information. And then we provide a number of tools after you sort of clean your data. We provide a number of tools to visualize and to analyze those data sets. And we're going to start with inputting the data, and then we have sort of in a similar fashion to what Ivo has demonstrated in other SOCR tools before. We have this interactive Excel-like, spreadsheet-like data visualization, data representation, where you can copy-paste your own data set. You can go to any table that you have and just copy-paste here. We are going to do demo using some of the predefined data set. SOCR also has a number of data sets that we provide along with the educational activities. I'm not entirely sure where the link was to the data sets. Or it's on. Do we have it here? No. Data sets, sorry. So we have a large number of data sets from, climate change, population, neuroimaging, biomedical, healthcare, census elections, all kinds of various data sets. So we are also incorporating many of these data sets inside of SOCR, so people can learn about tools and use some of the predefined data sets to sort of explore what capabilities we are providing. And I'm just going to start with data set that probably is familiar to many of you, and then we're going to try a couple of other ones later. We're going to start with iris flower data set. That's a classic data analysis and machine learning sort of data set that contains information about iris flowers, and we have 4 variables: sepal length, sepal width, petal length, petal width, and then species, and there are 3 different species that we're gonna have. And this data set, of course, is curated, so it has a nice range of values, nothing weird going on. All the species are there, no missing values. So basically, we can open Data Wrangler, and in this view, it shows this green bar indicates that there is no inconsistencies. It says 150 valid values, so there are no missing values. There are no weird strings or anything going on. They're all numeric. We can do various things with this. We can, as you click, for example, a column or a value, it sort of or a row, it sort of suggests you different things you could do. You could delete all the rows where the petal length is 1.4, and then it will do it throughout the whole data set for you. So it gives you intelligent sort of suggestions what kind of pre-processing or data cleaning you could do. You could just delete the row. You can actually, for example, drop a column. So now we have only 3 columns left, and if you go see, it's consistent throughout different parts of SOCR. So the data representation is persistent across different applets that you're using. Well, let's return the column. So what we're going to do, because this data set is sort of structured, well-structured and clean, we're just going to go directly into visualizations. And this is a very nice visualization that allows you to explore relationship between variables. Actually, we're going to do the histogram first. So if you want to look at univariate visualizations for just one single variable at a time, you can use something like a histogram that shows you accounts of specific values across the value range. For example, here we have sepal length, but you also can look at petal width. And then using the slider, you can regulate the number of beans that you have in your histogram. So the granularity of your representation is controlled interactively. It also gives you a summary information about each group or about each bar that you have in the chart. So you can look at median values, mean values, how many counts are in each group. So if you're looking at specific just one variable, you could use something like histogram. Trailers chart, on the other hand, provides the way to look at variable relationship. And then you can, there are a couple of cool features that we have here. First of all, it does color code by species. Second of all, it's interactive, so you can kind of rearrange that as you want, and you can zoom in and you can start. If, for example, if you're interested in this versicolor virginica types, you can just zoom in and look at the distribution of the relationship between them. How sepal width of one class is related to sepal length related to sepal width of the other class. And then not only we could look at 2 variables, we could look at large number of variables. So we can select 3, and then we can navigate sort of this 3-variable feature space. And then the same way interactively look at the different distributions, relationship between variables, or 4. So basically you can go as many variables as you have in a data set, and then we automatically add this scroll bars that you can go and look at all the combinations of the variables that you have that you're interested in too. So that's a nice multivariate visualization for quick exploration of the relationship between your measures. We provide a number of other standard charts. For example, the bar chart shows you, bar chart shows you the counts for specific sepal length related to petal length. This is a little bit of older version, but basically it does show you the mean value indicated as a red line here. And then we also have, in the newer version of SOCR app that's going to be released soon, we also have a couple more controls that allows you to normalize by mean, by standard deviation, some of the other newer options. And one other standard chart, such as scatter plot. So this is, again, just looking at the relationship. If you want to really in detail look at the relationship between just two variables, instead of looking at four at the same time, you can use scatter plot. So there are more standard stuff. Bubble chart allows to look at relationship between four variables at the same time. So for example, 3 numeric variables and 1 categorical. So the color is categorical, and then the X, Y, and radius are numeric variables. Pie charts, if you want to look at the distributions between distribution of, again, single variable, or for example, species, it shows you that they are all equal, 33% for each class of the flowers to have equal representation. Normal distributions try to fit a normal distribution curve into the distribution of variable values. So you can try that for all the numerical variables that you have in the data set. And I'm not going to go through every single one of those. Instead, we're going to just go into tools. And there, I think first what we can do is power analysis. So we do provide, for example, a number of power analysis, statistical analysis tools. For example, we can do a 2-sample t-test. So what we want to do is, for example, we want to compare the means of sepal length of 2 different classes of flowers, or 2 different species of flowers. So we're going to use the data. We're going to choose a specific variable that we want to compare across. We're going to choose the categories of species, and then, for example, we're going to compare first and third. So it does show you that there is a distribution of values, and there is a clearly they're not the same. And then given the sample size, the mean values, standard deviations, it computes the t-score and the p-value for this test. So you can sort of interactively perform mean comparison for the distribution of different variables here in the data set. And then also there is a lot of criticism that people often report p-values, especially in biomedical sciences, but they don't report sample sizes or means or standard deviations or the power of the test. And we have all of this value right here, so you can see that your power and then your confidence level parameter is reported right here. And then as you change your confidence level parameter, it updates automatically the chart. If you are done using the real data, you can also use it in a simulation mode where you can actually change your sample sizes. You can actually say, I want 100 observations here, and then what happens? And then you can change your means and standard deviations. Right. Yeah. Yes, exactly. And then there are no any other online web-based software that provides you these capabilities for using both with your data and in a simulated simulation mode, sort of. Another cool analysis that we have, and we can look at, is interactive clustering. So this allows you to perform k-means clustering on different kinds of data. So again, we have data set present here, so everything here is true. We select variables that we want to cluster against. So it does show you the projection on a 2-variable space. So X and Y, you have sepal length, petal width, you can change it. It will automatically update the chart. And then you want to choose the labels, species. So that color codes again as before. And then as you run your k-means clustering, it automatically interactively updates the results, and it shows you how it fits the clusters. So if you are users of k-means and are familiar with that, you know it depends a lot on the parameters. So you got to guess the number of clusters, and then you can choose the different distances that you want to use between the points in a high dimensional feature space. Also depends a lot on initialization. So usually people would rerun this multiple times to see sort of how it converges. And here we have the detect k. If you know the true labels, you can sort of try and see how well clustering results fit the actual true labels of the data set. So here, for example, we can see that the blue data set is better separated from the orange and green. And then we saw that before in the visualization when I was showing that there are two classes that are closely related, and another one is more different from the other two. So this sort of gives you the idea that what sort of kind of insight you can get from these data sets. No, you have to rerun it. Yeah, we can. Let me actually do another example. What we're gonna look at is a simulator SOCR knee pain centroid location data. It's data on knee pain locations that was simulated from real data. So the real distributions were used to sort of produce a larger amount of observations. And then this data set is actually fairly large. It only has three variables: x coordinate, y coordinate, and then view, which is a 4-value categorical variable. But it has a large number of values. Remember what was the total? Over 8, almost 9,000 values. So when you go to this size of data sets, the processing in the browser gets a little slow, especially if you do something like clustering or any other calculations that involve, I don't know, matrix multiplication and some other heavier operations. So it gets a little slower. Even charts get a little slower. So SOCR automatically detects that, and it suggests you that data set size may be a little large for wrangling and charting and some of the analysis. You still can do it. You'll just have to wait a little longer. But as an alternative, it suggests you to subsample the data set to some empirically estimated number of rows. So here, for example, it just suggests you uniform row subsampling. We have some sampling techniques in the development that would allow you to take into account your classes or some regarding a condition on some variable that you have. But right now, this version does uniform random row subsampling. So we can subsample. Now we have 4,000 rows against 3 columns, and that is a little lighter version of the same data set that we can analyze against. So we're gonna go and do clustering again here. I'm gonna choose both variables. Yeah. So as you can see, the simulated data actually has a very nice structure in it, because the idea here is that simulated knee pain, this is knee areas, left, right, and these two are front and these two are back side, rear side of the knee. And these are pain locations that are simulated from the real distributions. So since these have a very nice structure, you could sort of expect that some of the analytical tools probably will be able to capture that, and now we can try and see how well we can do with clustering. And at first, of course, if you use your labels, you can color code. So right front, left front, left front, right front, left rear, right rear, something like that. Then we can choose, say, 2 clusters. If you want to, for example, you want to cluster the front ones against the back ones and run the clustering. This Not always. So the bad initialization not always works from the first time. That's the key means part comes in. But then you can see, okay, we ran it once and it converged in this fashion. So didn't exactly separate the front and the back. And then we kind of may start thinking about why this could be happening. So one of the reasons I can suggest right away is that in the front distributions left and right, the density of the points is higher than in all of those in the back, either left or right. So that sort of drags the centroid. If centroid was located somewhere nearby here, then it drags the sort of the central mass of the cluster towards that area. Let's rerun again and see what happens again. Yeah, so it converges to this sort of to this condition when you have left and right in the front separated better than the front and the back. So this type of visualization gives you an intuition about how algorithm works, what can go wrong, and then how easy it is or hard it is to use, for example, these type of clustering algorithms to analyze this data. Let's switch to 4 clusters. Try to run for 4 clusters and see. Again, so k-means depends a lot on the initialization, and then since there were 2 clusters that were originally initialized in this orange area, they stayed there. So there was not enough difference to sort of pull them apart, or not enough points from the other back clusters to move the cluster center towards here. So we have here 1 correctly clustered type, and then 1 split into 2, and then 2 joined into another marker. And try again and see. Okay, similar situation again. One of the classes got split between 2, and this is the first time, first one this time. Again. So you can get the idea that this is not an easy problem. This is a hard problem, and then these type of algorithms are not always capable of easily to pick up these type of patterns. I can tell you that from my practice, especially when you're preparing the paper, playing with the parameters, and then we can actually try Mahalanobis distance. So Mahalanobis is kind of like Euclidean distance, but it does take into account data structure, so it uses the correlation matrices. So sometimes this works better. For our purposes, I was able to get almost correct separation. Yes, so something like this, right? I think we are getting. This is closer to, so using Mahalanobis distance that takes into account data correlations between data points performs a little better. So we get 2 correctly classified clusters, and then there is a little bit of confusion here, again, possibly because we subsampled some of the data, we are missing some of the observations, we lost some density points in the rear side clusters. If there are maybe the full data set, it would be a little better, but in this case, we got some of the points were misclassified. So we were able to perform on this specific data set an ideal clustering, but then again, it depends a lot on initialization. So you have to run it multiple times and see to what results it can converge. We have some of the other tools that are specifically used for some other problems. So we have survey reliability tools, statistical analysis. I'm not sure if I have data sets. Yeah, so this is a server version that you can run right now if you go to SOCR and then, if you go to SOCR main page and then if you try it online, you can run specifically this version of SOCR. We are currently preparing a new version that will be released very soon, and that has a lot more tools in them. and that has, for example, that T-SNE visualization that Ivo was demonstrating before with brain morphometry images. We will have that for your own custom data. So you can plug in your data and run a T-SNE visualization, for example, on it. So yeah, if you're interested, you can follow our repository on GitHub. All SOCR apps, including SOCR app, are on our GitHub. So you can go there and you can start our repository. You can watch, and that way you'll be notified when the new version is coming out. That's it for now.
Take maybe a couple minutes. Well, in the interest of, want to stretch? All right, let's take a couple minutes to stretch, and then we're gonna continue.